1 minute.
So while presenting, do I
Yeah.
need to like switch on the video, or I can stop up, right?
No issues. It's not compulsory. It's your wish. You can turn off your video or you can keep it, no problem.
Hmm.
So how about your classmates? Do you have any touch with your classmates?
Yeah, I think Anudeep is into software now.
Anudeep,
Most of the
which
people are
Anudeep?
into most of the people are into software, sir.
Which Anudeep? Shorter one or long one?
Both Anudeep.
Both are in software and okay.
Sir, both are in software, and Anudeep in between worked as this social welfare assistant in AP state government.
Okay.
And then he got shifted there, and after that, I think VRS has finished her MBA, and now she got married, and she's settled in Bangalore.
And how about this Ranjita? Ranjita got connected last long back.
Yeah, that is
If
what
possible, you try to
VRS
you
Lakshmi.
try
So
to add her in Alumni WhatsApp group.
okay.
I think she did his masters in business. I think.
Yes, sir. She has finished her MBA, and now she got settled in Bangalore. She got married.
Okay, Bangalore. She's staying at Bangalore. Okay.
Yes,
I
sir.
think he did. He did her degree in Christ University. I guess.
No, sir. She
She's
finished in I think IM. She finished. She did IM.
okay. Yeah. Okay.
Hmm.
How about others, Kowshik? Are there anyone in touch with you?
No, sir. We have a group like little bit of talkings and nothing else. And KK got married recently, sir. Like 1 week back or so.
Krishna Kishore. Okay.
So,
Okay,
however,
Kowshik. Nice talking to you.
sir.
So let us start the session. I request Dr. Nadeem to introduce today's resource person. Then he can start his discussion over the monoclonal antibodies.
How is Ujjwal, sir?
Nadeem, your voice is not audible. Once check your audio.
Am I audible now?
Yes, sir.
Yes. Yeah, you're audible.
Oh, okay. Good morning, everyone. It gives an immense pleasure to introduce today's resource person, Dr. Kowshik, who is presently working as a scientist at Lazuline Biotech Private Limited, Hyderabad, who is a proud alumni of our institute, KLU, and the Department of Biotechnology. He has done his BTech Biotechnology from our institute, MTech from Lovely Professional University, Punjab, and then he has done his PhD from NIT Warangal, a prestigious institute, and he got his degree in 2021. To his repute, he has published 8 publications in SCI journals, and he's strong at restriction digestion, ligation, PCR primer design, and bacterial transformation experiments and cloning. I have, Dr. Kowshik, with your permission, I have reduced your CV to 4 to 5 lines so that you can spend more time on the delivering of your monoclonal antibodies lecture.
No issues, sir.
And thank you for your valuable time.
Yeah.
I'll
Shall
hand
I
over
start?
you to. Yeah. Please go ahead. Thank you.
Just a second.
Yeah, sure.
Hope you can see my screen.
Yes, Dr. Kowshik, it's visible. Please go ahead.
Now this is fine, right?
Yeah, it's visible.
Yeah, just a second. Hello everyone, good morning. This is Kowshik and I'm here to discuss about monoclonal antibodies and their industrial production strategies. Am I audible to everyone? Is it fine?
Yes, Dr. Kowshik, you are audible.
Yeah, thank you. This lecture will give insights into the following topics like what is antibody, let us have some basics about what is antibody and what are polyclonal antibodies, what are monoclonal antibodies, what is the necessity of these monoclonal antibodies and why are they important to the industry, the production strategies, scope and future directions. And after that, one second. Yeah. Before going into the topic, let us see some basic concepts. What is an antibody? They are antigen binding proteins secreted by B cells. They can be membrane bound or secreted. Antibodies are produced by B lymphocytes after their differentiation into plasma cells. They are most important components of humoral immune response. They protect the host against infections. Production of antibodies is the hallmark of adaptive immune response. They are produced either to neutralize or eliminate antigens or pathogens from the body. These antibodies produced in response to a particular antigens are heterogeneous. So they are not monoclonal in such. So when it is in the body or the system or any other host, they are polyclonal. So it is also called as, they are also called as anti-sera. And let us see about the structure of immunoglobulins. How the immunoglobulin structure is elucidated. Several experiments were carried out to determine the structure of immunoglobulins. In 1939, Tiselius and Cabot confirmed that immunoglobulins were globular proteins present in gamma globulin fraction of serum. It took almost another 20 to 30 years to determine the whole structure. The credit of elucidation of immunoglobulin structure goes to Gerald Edelman from US and Rodney Potter from UK. Gerald Edelman. This is what the Edelman's experiment. And Edelman, he treated rabbit immunoglobulin with reducing agents such as DDT, iodoacetamide, SDS, and beta-mercaptoethanol. After the treatment, Edelman has separated the digested immunoglobulins by size exclusion chromatography, and he found two protein peaks in equimolar ratios. So these two equimolar ratio peaks has showed a molecular mass around 50 kD and 23 kD. So the 50 kD he called it as a heavy chain and 23 kD he called it as a light chain. And after this, Porter was working on proteolytic digestion of immunoglobulins using papain enzyme. He was using this enzyme to digest immunoglobulin in the presence of some other reducing agents. This resulted in three different fragments. As you can see here, this papain enzyme digests here and there are three different bands. Two portions with antigen binding capacity and one with crystallization capacity. The two antigen binding portions are identical and they also maintained antigen binding ability. They are called F antigen binding, FAB. FAB is called F antigen binding. The third fragment crystallized upon the storage and hence it is called F crystallization, fragment crystallized. And he also observed that the ratio of FAB to FC was found to be 2:1. After that, the Nisonov experiment. He used pepsin to digest IgG and it yielded a 100 kD fraction and numerous small fractions. The 100 kD was double the weight of the Porter. It is bivalent. It has only antigen binding region. And based on the above experiments, the structure of antibody is revealed and it has the following features. All the antibodies share a common structure. It has two heavy chains and two light chains. They are heterodimers. And after that, the light chains are of molecular weight 25,000 Daltons and heavy chains are of molecular weight 50,000 Daltons. So coming to the structure of antibodies, light and heavy chains were bound by intrachain and interchain disulfide bridges. The exact number and precise positions of these interchain disulfide bonds differ among antibody subclasses. C refers for constant region and V refers for variable region. The first 110 or so amino acids of the amino-terminal regions of a light or heavy chain varies greatly among antibodies of different specificity. There are 5 different classes of antibodies: IgG, A, M, E, D. So coming to the polyclonal antibodies are the antibodies that are produced in the serum. They are also called as anti-sera. The steps involved in the production of polyclonal antibodies are: first we have to immunize the organism, and then we have to collect the blood, and then the antibody has to be purified and they have to be identified. These polyclonal antibodies are also known as anti-sera. They are the products of numerous B lymphocytes. Animals are the source of polyclonal antibodies. Polyclonal antibodies can be produced by in vivo, which means the requirement of animal is essential. So this makes it a pitfall, because you cannot kill so many animals. So we have to go for monoclonal antibodies. Production as well as repeated production of polyclonal antibodies requires booster injection. Polyclonal antibodies production does not require highly skilled manpower. Sophisticated instruments are also not needed. Sensitivity vary. Polyclonal antibodies are not homogeneous, as they are heterogeneous. Coming to monoclonal antibodies, they are, as the name indicates, they are produced by a single clone. Monoclonal antibodies have various applications in diagnosis and therapy for several diseases such as cancers, autoimmune diseases, sexually transmitted infections, and others. In recent years, the use of monoclonal antibodies has been expanded due to significant advances in design. So these are the different types of antibodies, monoclonal antibodies, that are produced. This is fully human antibody, and this is humanized antibody, which has the C and variable regions and light chains, and this is chimeric, and this is murine, that is produced from mouse. The nomenclature of the antibodies is given like this. The first, it has a prefix and 2 infixes and stem or suffix. The infix 1 in the name of monoclonal antibody is meant to provide the information about the target of an antibody. This is the reason that infix 1 is usually directed towards the target or the disease class against which it is effective or targeted. USANC has approved the list of syllables that are specific for the disease or for the target. And coming to the infix 2 is meant to identify the source of antibody. The infix 1 is to provide the information about the target of the antibody, whereas infix 2 is meant to identify the source of antibody. Certainly for therapeutic applications, monoclonal antibodies, the source information is crucial, because safety is important and we have to understand from where it came, from where it is origin. And after that, additional words are also used while naming monoclonal antibodies. They are typically used as clarifying words for the nomenclature of monoclonal antibodies that have various payloads. This is what you can see the table. The infix 1 determines the name of the antibody that is targeted, and infix 2 the source, and suffix 1 is MAB. So you can see that, for example, rituximab and adalimumab likewise. So coming to the industrial production, we have hybridoma. In order to produce the monoclonal antibodies, we have different technologies. The first one is hybridoma. Hybridoma technology and fast display technology are used to design monoclonal antibodies, and these are culture production factors. For example, the culture production factors include cell lines, culture medium, culture conditions, production platforms, and production systems. Coming to monoclonal antibody production, these are produced by hybridoma technology. Monoclonal antibodies are produced by hybridoma technology. The first step involves immunization, and the second step involves preparation of splenocytes. In mice, a very limited volume of peripheral blood can be drawn, so which can become a limitation. So now we have to think for the alternative source. There we can produce more and more antibodies without animal source, and therefore the best source is lymphocytes, is spleen. Dissection of spleen and preparation of splenocytes has to be performed under aseptic conditions. After that, the cells has to be fused. Why the cells has to be fused? Because these cells, the splenocytes, have limited number of generations. That's what they die after some generations. So they have to be fused with a cancer cell, which makes them immortal and can produce continuously. So the fusion of cells involves the fusion of splenocytes and myeloma cells. They are mixed together in a specific ratio. A cell suspension of the mixed cells has to be treated with agents that can induce fusion. So these agents include polyethylene glycol, Sendai virus, etc. Nowadays, the cheapest method to fuse cells is by using polyethylene glycol. After the fusion of splenocytes and myeloma cells, these cells are very much fragile. So they have to be addressed, or they have to be, next experiments, all the further experiments has to be performed with utmost care. How to select, after fusion is done, how to select the hybrid cells? When a mixture of fused cells is grown in a hypoxanthine, aminopterin and thymidine medium, so HAT medium, the aminopterin works as a folate antagonist and blocks de novo pathway for the synthesis of nucleotide. In the due course, primary cells will die because they have a limited lifespan and they cannot synthesize DNA. Myeloma cells that do not get hybridized will die. And after that, only those are hybridized will get propagated in HAT medium. After that, the selection of clones and the expansion. The selection of clones fused, he has to select. The experiment involves plating of cells in 96 well plate. The experiment is designed in a manner that after dilutions, each well contains a single antibody producing cell. Expansion of clone is done after the identification of a specific clone. It has to be expanded so that it can be used for larger antibody production. There are two different methods that are there for the expansion of clone. One is in vitro method and the second one is in vivo method. After both the methods, these monoclonal antibodies has to be purified. They can be purified by different methods. For example, you have affinity chromatography and the culture production factors. What's it?
Nothing.
Yeah. To produce monoclonal antibodies, we need a cell line. There are different cell lines that are used to produce monoclonal antibodies. So for the best production of monoclonal antibodies, cell lines choice is very much crucial because these cells must be stable and secrete the desired quantities. Choice of cell line is pretty important for monoclonal antibody production in the industrial scale. The mammalian cells are the most commonly used expression systems for monoclonal antibody production, because if this expression is considered in yeast or any other bacterial systems, bacterial platforms, we don't have glycosylation, and there is the problem of forming aggregates or denatured proteins or truncated proteins. So mammalian system has an advantage that cellular machinery is adapted for the production, processing and selection of monoclonal antibodies. Out of all those cells, the most commonly used cellular platforms, mammalian cell platform is CH4 cells, Chinese hamster ovary cells. Genetic modifications in CH4 cells have generated cell lines capable of producing like high quantity of humanized, around 100 picogram per cell per day. Coming to this cell culture, the second cell culture production factors, culture medium is very crucial. Cultivation media for mammalian cells must have a complex content of ingredients ranging from amino acids to trace elements. Each and every element that is required for the growth of monoclonal antibody producing cells are very important. To supply the cellular demand, the culture medium uses serum in its composition. However, due to the emergence of diseases caused by defective prions or any other bacterial things, there is a great incentive to remove any animal component from the culture media. Especially if the medium is used for industrial production or biopharmaceutical products, we have to be very careful while using the serum. The media should be animal origin free, because if it is of animal origin, it can be prone to contamination more. Optimized media is very important. So now the development of a proper medium can be time consuming and very expensive. So many companies prefer to design the media on their own and maintain the composition between production lots, as well as to develop appropriate medium composition for the specific cell type that will be used. And it has to be considered that these culture medium are very important, because to get the better productivity, the development of downstream process is also very important. And coming to the culture conditions, growth conditions can directly influence the cell growth, because these are the conditions that has to be maintained to give an optimum environment for the cells to grow. So usually mammalian cell cultures for monoclonal antibody productions are very well defined. The temperature is maintained at 37 degrees Celsius. and the pH is around 7 to 2, 7 to 8 or 7 to 7.5, and the dissolved oxygen level should be around 30 to 60%. CO2 level is monitored to conditions have shown great potential, and the CO2 level should be around 31 to 54 mmHg. Bioprocess can be designed to occur in two phases. The cell growth is optimized to reach certain cell density. After that, once the density is reached, the second phase and the bioreactor conditions are shifted so that the cells continue to grow just at a maintenance rate. Some CHO cell strains, as we have discussed, the CHO cell lines, they are crucial or they are mostly used. These CHO cell strains and hybridoma cells are sensitive to changes in temperature and pH. So the culture conditions are pretty important. Temperature, pH, each and every cell have their own temperature, own pH, and optimized dissolved oxygen as well as CO2 levels. For example, coming to the CHO cells, the pH should be maintained around 6.7 to 7, and the temperature varies between 30 to 35 degrees Celsius. The growth metabolism reduction also contributes to lower production of some metabolic compounds which are toxic for the cell, because when you don't maintain these conditions, the cells undergo stress and may produce secondary metabolites which may be toxic to the remaining cells or which may be toxic to the environment and can degrade cells or make the cells reach dead stage. And a good way to monitor the growth stage of cell culture for controlling changes in cultivation is watching Q and CO2 levels. And coming to the production platforms, there are three different production platforms that are used. They are batch, fed batch, and perfusion cultures. The cell culture for monoclonal antibody production can follow three of the different types, any of the different types of the process. The simplest of them is batch production. In this, this has a closed system in which bioreactor is sterilized, prepared with a medium containing all the nutrients needed for the cellular growth or withdrawal of spent medium. As the process runs, the nutrient concentration decreases and waste metabolites are produced, lowering cell viability. In spite of being a simple process, batch is not the most suitable. It is not the most suitable because it accumulates waste metabolites, it degrades more sensitive products. So cultivation factors such as initial nutrient concentration and waste metabolite production directly determine the maximum concentration of cells that can reach. So this type of cultivation, for example, in batch cultivation, the cell density is around 1 to 2 into 10 power 6 cells per ml, and then cell viability drops because of the accumulation of metabolites and nutrients. And the production process lasts for 4 to 7 days. When the productivity reaches a certain concentration of interest, supernatant is collected and the downstream process is continued. In contrast to a batch, the second type of production process utilized is continuous fermentation. There are two types of continuous production, chemostat cultures and perfusion cultures. Concerning chemostat cultures, fresh medium is added to the bioreactor and fermented medium is removed along cells at a constant flow rate. So that's what it is called chemostat. So the culture volume remains unchanged. The flow rate controls cellular growth, and when these two variables are equal, the bioreactor reaches equilibrium. So here in this context, fed batch, the cells may reach to a density of 10 to 3 into 10 power 6, whereas in batch 1 to 2 into 10 power 6 cells per ml. So compared to batch, fed batch has more cell density. To avoid viable cell losses, along with the constant outflow of the byproducts of cell metabolism, many manufacturing plants have developed cell recycling system, and thus the perfusion culture method was developed where the cells kept inside the bioreactor. The disadvantages of continuous fermentation are the use of large amount of expensive culture media and the difficulty in recovering the product, which becomes out fairly diluted. The accumulation of, and these are also the disadvantages of accumulation of waste metabolites, degradation of more sensitive products. And coming to the third type of processes of producing monoclonal antibodies is far by the most utilized at industrial scale. Perfusion cultures are most utilized at the industrial scale. In this process, the cell density reaches 8 to 12 into 10 power 6 cells per ml. Cells are maintained in relatively optimal biochemical content, whereas the cell culture reaches high density and high production. Although monoclonal antibodies produced by fed batch processes, there are tendencies indicating in future many bioprocesses. Why perfusion cultures are not opted mostly? In most of the industries, they opt for fed batch cultures, whereas in perfusion cultures, why it is not opted is lack of homogeneity in the continuous reactor vessel and challenges regarding long-term operability and maintenance, high cost and long times required for the process development experiments, and genetic instability of the cells is also a drawback for perfusion culture systems. Coming to the production systems, normal production systems, the use of monoclonal antibodies as therapeutic drugs requires a large scale production. The normal scales are not sufficient to meet the requirement of the production. So various production systems have been developed and they have evolved while new alternatives are emerging. The production of monoclonal antibodies at a commercial scale can be performed with adherent cells or suspension cells, although the latter is far by most used and is better established with the more efficient production available for cells cultivation. Thus, scale up using suspension cells is easier. Another advantage of the suspension production system is that a bioreactor with a large area for cell adhesion is not necessary since the cultivation of adherent cell productivity is directly linked to the bioreactor's area. Here, the company GeneTech has been producing Avastin, Herceptin, Lucentis, and Rituxan, also called as Rituximab, in 96,000 liters bioreactor. And after that, Bevacizumab, addition of 90,000
Kowshik.
liters has been performed in 2007. And whereas 2000 or to 2009, this 1,40,000 liter has been 2 lakhs has been upgraded to 2 lakhs liter system. So now they are producing at a very large scale, which is far from belief. And Abai Technologies have Humira, the adalimumab. They are producing it in a scale of 32,000 liters from 2007 to 2013. Now they have upgraded to 56,000 liters, and by 2024 they will be upgrading it to 1 lakh liters. And these are the reactors used. Coming to the production systems and the different types of bioreactors commonly used for monoclonal antibody production. They are stainless steel bioreactors, airlift reactors, disposable bioreactors. Stainless steel bioreactors. This is the first one which is showing the stainless steel bioreactors, and they are stirred tank bioreactors, and this is the most consolidated type of bioreactors for monoclonal antibody production. They contain baffled stirred tanks, and the cultivation in these bioreactors allows for a wide flexibility of working volumes ranging from 1 to 25 liters. These are large scale reactors since the system is easily scalable to larger volumes due to its higher control. Initially, the research and development R&D will be conducted in 3 to 5 reactors, and this 25 liters will be a pilot scale, and in the large scale this can be easily scaled up. And the mechanisms and cleaning and sterilization protocols for this are well defined, because from age old many industries are using these kind of bioreactors. So all the process is well defined. All the process is well defined. And additionally, cultivation parameters for the system such as gas transfer coefficient, agitation, aeration, temperature maintenance, pH, and others are well controlled and regulated when compared to other production systems. Another advantage of stir tank reactors is that it can be used for the cultivation of various cell types in addition to the products obtained from the cultivation in this type of bioreactors. These are easily approved for therapeutic use as regulatory terms are well defined for this kind of production. Airlift bioreactors. Airlift bioreactors. Airlift reactors are also broadly used for the industrial production of monoclonal antibodies. These reactors, this is the airlift reactor. These reactors consist of a tank with bubble column inside, and air is injected into the column base. The air flows, you can see the arrow mark. The air flows through the column's length to the top of the bioreactor as degassed culture medium flows in the opposite direction. This creates a constant gentle mixing of the medium as well as proper culture aeration, annuling part of the shearing stress caused by shearing systems. And other advantages of this operation system are that it is easier to scale up, contamination problems are less, and the equipment is simpler. In spite of these advantages, this system is less utilized than STR stir tank reactors, as the working volume ranges only from 2 to 5 liters. And the airlift reactors handling is not so easy, and it is not clearly elucidated. Coming to the disposable reactors. These disposable reactors, the first single-use bioreactors, these are the single-use bioreactors. The first single-use bioreactors emerged in the late 1990s with the launch of wave reactor system. After that, disposable stirred tank bioreactors were developed. This method brought many advantages for monoclonal antibody manufacturing. At the end of the process, the bioreactor is discarded and replaced by a new clean and sterile one. This eradicates cross-contamination between batches and decreases the time consumed with the equipment. In these kind of bioreactors, if at all you need to use different cell culture platforms, these systems, as these are single-use bioreactors, you can discard the things, and hence these are mostly preferred if you have different cell lines to produce different monoclonal antibodies. And this also decreases the time consumed with the equipment preparation between the batches. You need not prepare the equipment like sterilization in place, cleaning in place. You need not go for these kind of things. Directly you attach a bag and start doing it, and once you're finished, you discard the bag. When having all the advantages of this process taken in an account, the savings are made regarding production, because you have to have less amount spent in cleaning and steam and all these kind of things. And for this kind of bioreactors, the investment capital is highly significant when compared to other process methods. The great disadvantage of this production system is small work volume supported, ranging from 50 to 2000 liters. You have disposable bags, disposable stirred tank bioreactors at a range of 50 to 2000 bioreactors. The wave system consists of a sterile plastic bag. So you can see here, this is a sterile plastic bag lying on a rocking platform. The bag is half filled with the cultivation medium and half filled with the gas mix. So these are the sensors for PO2, and you add half of the gas and half of the medium to maintain good interaction of these cell platforms. And the platform motion creates an undulation movement in the culture, ensuring efficient aeration and culture mixing without causing shear damage. The other available systems combine the convenience. For example, these are combined with some other systems like millipore, high clone, and XLRX, etc. The main features of these single-use bioreactors are related to the technical characteristics, like they are far more better than stirred tank bioreactors and also airlift bioreactors. SUBs are being widely used to replace many processes for the production of bioproducts. SUBs are cheaper and more efficient. They are more efficient alternatives when compared to these two kinds of reactors, that is stirred tank reactor and airlift bioreactors. SUBs provide an efficient way to deal with different kinds of products or different kinds of cell lines in a single shot. So, the necessity of monoclonal antibodies. Now we have seen the production strategies and production platforms and the culture definition and the cell culture conditions importance in designing a monoclonal antibody production protocol. So what is the necessity of these monoclonal antibodies? This hybridoma technology serves as an immortal source of monoclonal antibody. So they can produce they can go for n number of generations and produce continuously these monoclonal antibodies. The same quality of antibody is maintained. So as they are from a single clone, and the same quality is maintained amongst the different production batches. High reproducibility, scalable, and unlimited production source. As these are fused with the cancer cell, they are immortal and they can be a source of unlimited production. The speed and sensitivity and specificity of the assays, so they can be easily recognized, and they can be easily, like what to say, assays, they can be recognized with assays very easily. We can produce antibodies whenever they are needed. If at all a cell line is ready with a particular clone of your choice antibody, you can produce whenever you want it in these different kinds of bioreactors. For example, you have, if at all you wanted to go for a 25 liter fermenter, and you can go for stirred tank bioreactor. So no need to worry about maintaining the animals. The main problem with the polyclonal antibodies is you have to immunize the animal and you have to collect the blood. So there you have to kill so many animals. So which is not suggestible, because if at all you want a large scale production, you have to kill so many animals. So this is not the way. So now what we do, we immunize the animals and we collect the cells that start producing these antibodies, like particular antibodies, and then we'll go for hybridoma technology, and continuous production will be there. So now you don't have the option or need to kill many animals. And the antigen immunogen need not be pure. The selection helps to identify the right clones against the specific antigen. These monoclonal antibodies are against a single epitope of the antigen. So these are the monoclonal antibodies approved till now, and out of them the recently approved things are tezepelumab and tebentafusp and faricimab, satulimab. So the future direction, because the future always monoclonal antibodies has a bright future, because they are needed and designed as a target therapy. The product optimization is really needed. If one concentrates on the optimization of the production capability or the need of the production, so they can be a main player of the industry. The process need to be optimized to use lesser media in order to generate more and more antibodies. The productivity, to increase the productivity. So productivity increase, for example, productivity increase and optimization, and is very crucial for an industry, because it serves a big cost cutoff, and these target oriented therapy. So you have, for example, you're doing monoclonal antibodies for particular targets. So you can engineer antibodies and you can identify the sites, and you can modify by using site directed mutagenesis, in vitro site directed mutagenesis, like these kind of things. So any queries?
అయిపోయింది మీటింగ్.
Dr. Kowshik,
Sir.
thank you for your valuable time and your thought-provoking guest lecture. Now the forum is open for queries. Dear students, if you have any doubt, please ask to our resource person. Dear students, if you have any doubt, if you have any queries, please ask.
phone he take it.
Kowshik, can I have a couple of questions for you?
Yes, yes.
Okay. May I know the profile of your company? I mean, exactly what it is doing right now?
We have a flagship product is human albumin.
Human albumin.
Recombinant human albumin.
Okay.
And we are producing two cancer drugs like elasparaginase and pegasparaginase.
Okay.
We also have blood thinner, enoxaparin, like low molecular weight heparin.
Okay. Blood thinner.
Currently, we are working on these 4 products, and we are into the manufacturing of this insulin, Lispro, Aspart.
Okay. So, is there any opportunity for our students for as an interns for a period of 1 month or 2 months, something like that?
Yeah, we can have that, no issues.
Acha, how many maximum students you can accommodate?
I think 4 will be fine.
4 will be fine. Okay. That's
Because if there are too much in number, the concentration will be less on people, right?
Yeah, I understand, Dr. Kowshik. Yeah, students, it's your time now. Go ahead.
Hey.
See, I think Dr. Kowshik, students have learned so much and they don't have any doubts with your presentation. Okay. Dr. Kowshik?
Yes.
Yeah. Thank you for accepting our invitation and presenting a thought-provoking talk. Thank you so much.
Yes, thank you.
Thank you. Yeah, yes, Dr. Kowshik, you can leave. Thank you once again from the entire department and students.